Thank you very much. Thank you for the invite. Yes, so I'm going to be talking about MEG. There are 8 MEG labs in the UK shown here at the moment. And for practical purposes, I'm not going to spend a lot of time talking about the differences between MEG and EEG, but we've heard a lot about using EEG biomarkers in terms of ERPs and oscillatory responses. And MEG is a very similar technique in terms of the phenomenology that it can measure and the way that you can treat the data. We can, if somebody wants to buttonhole me at lunchtime and we can have an argument about the pros and cons of MEG versus EEG, I'd be very happy to do so, but I don't propose to do that today. So everything that I talk about with MEG, you could in principle do with EEG. So I always forget my acknowledgement slides, so I've started putting them at the beginning. There's an awful lot of people that have contributed to the data I'm going to show today, in particular Suresh at the top, who's recently left Cardiff, but he was the MEG lab manager, and some key collaborators on the right from other sites away from Cardiff. So I'm going to talk briefly in 3 subtopics. The first, I want to address the relationship between oscillations and the kind of things you can measure with fMRI. And I'm not going to spend a lot of time on this because in a way I'm preaching to the converted here, I think, because this is a call to try and move away from rather non-specific measures of neural activity, which, if you think about it, doesn't really mean anything as a term, to more physiologically based specifications of what's going on in the brain in terms of activity. And most people in this room already buy into that as a program anyway. But I think there's some interesting things I can talk about along the way. Then I want to talk about what other people have described as sort of using oscillations as biomarkers, which is trying to pick very robust, repeatable, and translatable measures that we can measure with MEG and EEG and tie them to synaptic physiology and pharmacological manipulation and disease modifications. And I'll talk at the end, if I don't run out of time, about biophysically informed modeling. DCM is dynamic causal modeling. So functional imaging, and I would put MEG and EEG into a very broad category with fMRI and PET as being functional imaging techniques. I know they're not technically imaging, but I'd like to put them in that category for this purpose. And whatever technique that you choose to use, we're trying to bridge the gap between the neuron and whole human behavior. And fMRI is a technique that I use myself, has fantastic spatial resolution, but we don't really know what it's measuring in terms of its link to the underlying neural physiology. And people often talk, and I've used this term myself in papers, about measuring neural activity and differences in neural activity between tasks and groups. But it's a very non-specific and ultimately not very helpful concept. So Fred had his scary diagram that he uses to scare his colleagues about EEG. This is what I use to scare my colleagues in the FMRI group at Cardiff. And on the left in blue are all the things that we'd like to measure and characterize in humans. So this is the psychology of human behavior, if you like. So it's things like perception, awareness, consciousness, memory, emotion. On the right is the complex hemodynamics associated with fMRI, giving you these beautiful pictures here. And in the middle is the kind of things that, as physiologists, you guys are interested in and measure on a day-to-day basis, which is the kind of things that are happening at the level of the synapse and the cortex and in the neurophysiology. And it's the kind of phenomenology that we can measure: gamma, alpha, beta, theta, delta, characterizing amplitude, frequency, phase. And at the top are all the things that modify these modulatory factors, such as drugs, aging, disease, structure, neurotransmitters, connectivity, development. So I also like to show this to my MSc students and tell them that it'll be in the exam. But I don't do that. That would be mean. So the complexity of the problem we're trying to address, if we want to do functional imaging and use it to bridge this gap, is we need to do more to understand this area. And this is about human neuroimagers such as myself talking properly to animal neurophysiologists about how we can use information from here to help us understand what we measure with non-invasive techniques. So in an understanding, for example, the fMRI signal, you can do simultaneous measurements. This is logothetis' work, doing simultaneous LFP recording and bold. At Cardiff, in my group, we have a different approach, as we do the same tasks with fMRI, and then separately, obviously, we do the same tasks with MEG. And we look for where in the brain we see oscillation, band-limited oscillatory power going up and down. It can either go up or it can go down, and we compare that with the spatial maps that you get with FMRI. And this is just two papers from my group using it's one paper from my group with two different tasks. The panel on the left is a visual task, and the panel on the right is a verbal fluency task where people have to name words with a given letter. And the top row is the group FMRI map. And the middle row is what you get if you do the same task in MEG, and then you do source localization and look to see where you have changes, significant changes in band-limited power. And what you actually find, and this is actually common for both tasks, is that wherever you get a bold response, at neural activity if you like, you get a suppression of alpha and beta rhythms. Now the MEG maps, and this would be true if you use, for example, Loretta with EEG, the spatial resolution is much reduced compared to FMRI, but you can see that there's a general correspondence. And if you pick out some representative slices, you can see that the suppression of low-frequency rhythms occur in the same places that you get the bold response. And in fact, there's been very neat invasive recordings in epilepsy patients, comparing them to bold responses in humans, which show that you have a kind of a seesaw effect. If you look at the correlation between bold fMRI signals and frequent and band-limited power in both the LFP and the multi-unit recordings, you see a negative correlation with low frequencies and a positive correlation with the higher frequency gamma signal. So why bother doing MEG and EEG? Well, in the grey, I've got the thing that we always tell our students, which is that MEG and EEG are really good because they have really high temporal resolution, a millisecond. But actually, the thing that I think is most useful is that MEG and EEG have a direct link to neuroelectric activity. So you're not seeing things through this blurred hemodynamic correlate. And this is where the power of the techniques lie in terms of trying to link us in a translational way to animal and in vitro studies. Now, amongst the kind of cognitive neuroscience and human and oscillatory MEG and EEG research community, there's an increasing model coming which separates oscillatory dynamics into local, interregional, and global measures. And it looks like, this is a simplification, but it seems to be a useful one, that very narrowband gamma, high-frequency gamma oscillations, which I'm going to talk about for the rest of my talk, mostly represent local processing in cortical columns. And then as you go up to interregional networks, say between V1 and V2 in the visual system, there's a big debate about the role of narrowband gamma oscillations, but there's certainly evidence that alpha and beta oscillations convey information in terms of connectivity between regions. And then as we go up to whole brain networks, these again seem to be mostly characterized in terms of modulations and coherences in lower frequencies like alpha and beta. So this obviously is probably way too simplistic, but it appears to be a useful way of thinking about things right now in terms of the state of the art of the literature. And I'm going to talk mostly about this local visual gamma oscillation. I'm getting ahead of myself. So in the MEG community in the UK, there are 8 of us, and we're actually coming up with standardized oscillatory protocols that we're now running in large normative cohorts. We've got a cohort that we're putting together of 640 people where we try and probe local cortical function in the visual system using a visual paradigm, simple motor paradigms to look at motor gamma and beta, ERD and ERS in the motor system. And then we're looking at characterizing resting state networks with MEG and the conventional eyes open, eyes closed alpha paradigms as well. So we're trying to come up with standardized and repeatable protocols that all sites are following right now. So for the rest of my talk, I want to talk about one very specific oscillatory signal, which is the visual gamma oscillation. And the reason I want to talk about it is that it out of everything that I've done in measured in my research career, it appears to be the thing that's most easily linked to animal neurophysiology. But there may be a bias there because I'm a vision researcher, and so I tend to look at the vision literature. It's very robust and repeatable, as I'll show you, in humans. It appears to give us a window on the excitation-inhibition balance, and hence maybe a marker of synaptic integrity with a particular focus on GABAergic function. So there's a literature coming out of the two groups in Europe in the 90s, Wolfsinger's group, and this is from Kaiser and Koenig about the same time, where they discovered that if you show animals, this is cat data, very simple visual stimuli, this is a grating, you get a very characteristic oscillatory response out of primary visual cortex. So this is a recording in V1, the primary cortex. So the stimulus comes on at 0, you get a broadband spike that goes from 0 to 200 hertz here. That adapts out very quickly. Then you get a sustained narrowband oscillation, which is the thing that I'm going to be talking about. This is the visual gamma oscillation. You also get suppression of the alpha and beta rhythm. And we can do those experiments in humans. We put people in a MEG system. We divide the time series into stimulus present and stimulus absent time periods. And then we do what's called the beamformer source localization. And if you do that, you can see, this is work from Matt Brooks in Nottingham. You can see that even to these very simple visual stimuli, you get multiple oscillatory effects. You get an alpha suppression in the primary visual cortex. You get gamma ERS, a gamma increase in power. And you also get a sustained DC change as well. And these all coexist with where we would see the changes in the bold response. So there's a rich and complex phenomenology that comes out of even a very simple experiment. And this is data from our lab. So we show people a grating patch. And what's really important about this stimulus is that there is actually no temporal content. It's passive viewing, and the grating isn't moving or flickering. So any oscillatory signals that you see in the time-frequency spectrum are generated by the brain itself in terms of some kind of frequency loop to generate that. So we do source localization. This is actually data from a single person. So you can do this repeatably and robustly in one person. We localize the gamma response to the primary visual cortex. Then we place what's called a virtual electrode at that point, do the time-frequency decomposition to show both evoked and induced effects. And you see basically the same signals that you see in the animal recordings. So you see an evoked response, you see alpha-beta suppression, you see a narrowband gamma, and you see a hint of a higher frequency spike at onset. Now, what I think if you take away one message from my talk, it's that when we do these experiments in humans using MEG and follow the analysis pipeline that I've just shown, we get a signal which looks almost identical to what you get from the LFP recording in macaque monkey. So this is a very, I would argue, translatable signal in terms of being able to understand it by comparison with the animal literature. And there's some interesting things that come out of that. So this is monkey LFP work from Ray and Maunsell, from John Maunsel's group. And they showed that there are at least two different gamma signals. There's this narrowband signal that we're characterizing in the MEG that I just showed you, which it's generated in V1, but it's not correlated with the firing rates of cells in V1. So it's not linked to individual cell firing. Whereas there is a broadband gamma signal where if you measure from individual units, the broadband power, the gamma in this range is somewhat correlated with firing rates of cells. So there's at least two different gamma signals in the visual system. And they appear to be related to different things. It's layer specific, so we know the visual gamma comes from predominantly from the superficial layer 2, 3, whereas the beta signal comes from layers 5 and 6. So there is a beta oscillation in the visual system as well. I'm running out of time, so I'm going to skip over this a little bit, but we can show that these differential gamma signals are modulated by higher level cognitive processes like attention. This is actually monkey data showing that the firing rate is increased by visuospatial attention, alpha and beta are suppressed, but gamma is, if anything, slightly reduced. The narrowband gamma is mostly unchanged, and with a MEG paradigm, we could show much the same thing. So in the MEG paradigm, when we do the version of the experiment and modulate attention, we can show that the alpha-beta signal is modulated by attention. It's suppressed. You get more suppression when you attend to a stimulus. The narrowband signal is unchanged, so it's only driven by the stimulus and not by higher-level cognitive processes. and the broadband gamma, which remember we think is more linked to the firing rates of cells in primary visual cortex, is modulated by attention. So these signals are differentially sensitive to behavioral parameters. Okay. That's what I just said. So one thing to say, if you put people in a MEG scanner and repeat the experiment every week for 5 weeks, you get almost identical time frequency signatures. So this gamma signal is very repeatable. And if you're a 43 hertz gamma responder today, you'll be a 43 hertz gamma responder next week. But in a year, you'll be a 42.5 hertz gamma responder. So you lose about half a hertz per year. It's highly heritable. This is a study from Pascal Fries's group where they looked at monozygotic and dizygotic twins, and they were able to show that monozygotic twins have an identical gamma frequency to all intents and purposes, whereas dizygotic twins are somewhat correlated and unrelated twin pairs have no correlation in frequency. So whatever is driving variability in gamma frequency, it's highly heritable. So the question is, what drives that? And we think it's to do with the way this inhibitory interneuron pyramidal cell network is actually constructed. So the gamma rhythm appears in the visual system to be essentially constructed in this canonical little microcircuit. And it's the properties of that excitation and inhibition balance within that circuit that set the network frequency. I'm going to skip over this a little bit, but my colleague Suresh has spent a lot of time giving people lots of different GABA modulating drugs designed to try and shift this EI balance around, and then looking at what happens to the gamma signal. And it turns out, I'll just pick one actually, if you give people alcohol, 6 vodkas as it happened, you actually boost the gamma amplitude and shift the frequency downwards, and that's consistent with the modulation of the GABA system with alcohol. Similarly, if you give people propofol, you boost the gamma signal. If you give them ketamine, you boost the gamma signal. And I'll talk about tiagabine a little bit later. So I want to finish, I know I'm running out of time, by just talking a little bit about modeling and why modeling is important. And this is work with Rosalind Moran and Carl Friston. Basically, what we do is we take the data from the MEG that's been source localized to primary visual cortex to give us these gamma oscillations, and we put them into a model, a neurophysiologically informed model. This is the model. Now, for people like myself, this is a very complicated model. For people who actually know about the neurophysiology of the visual system, this is a ridiculously oversimplified model of what's going on. But there's a deep layer of pyramidal cells, there's a layer of spiny stellate cells, and there's a layer of superficial pyramidal cells. And then there's a population of inhibitory interneurons, and there's connections, forward and backward connections between the populations. And where the connections exist, that's because in the animal literature, these connections have been shown to exist. And where a connection doesn't exist, so for example, there's no connection back from the superficial pyramidals to the spiny stellates in the visual cortex, that's because in anatomical studies of primate V1, there isn't such a connection. Now, the reason that I've labored that point is this is a way for myself as somebody who only does non-invasive human work, it's a way for me to take information and literature from animal neurophysiologists and build it into my model so that I can then help inform my analysis of my data. So it's a way of uniting the two domains, if you like. And when you do that, I'm going to skip to because I've run out of time. You find that certain parameters have very strong control over the gamma frequency and amplitude. So for example, in particular, the network frequency, gamma frequency, is set by inhibitory gain control on the superficial pyramidal cells. It's also set by, as you might expect, by the time constant in the inhibitory interneuron. So the longer the time constant, the lower the frequency. And the gamma is basically dependent on any kind of inhibitory connections that are associated with the superficial pyramidal cell. So a very important one is that the amplitude is set by the strength of the inhibitory connection up here between the inhibitory interneurons and the superficial pyramidal cell. So the more inhibition there is from the inhibitory interneurons, onto the superficial cells, the higher the gamma amplitude. Okay, so if there was any kind of disruption of that link, or if you had reduced inhibition in a disease state, you'd expect to see gamma amplitude reduced. And we do have pilot data showing that in schizophrenia patients, it's precisely this connection that is impaired in the data. So we see a reduction in gamma amplitude, and when we do the modeling, we see that there's a specific reduction in this inhibitory connection. Now, the model might not be right, may be far too simplistic, but it's a way of starting to incorporate neurophysiologically informed modeling into our analysis of the data. And also, you should be able to see how you could do this in pharmacological studies. So we put this same analysis onto a drug called tiagabine. Tiagabine is a drug which boosts the amount of GABA available at the synapse. It's a GAT1 reuptake inhibitor. Blue is placebo. So in the placebo sessions, you see no change in gamma frequency. This is gamma frequency. And then as soon as you give people a drug, at the 3-hour time point, the gamma frequency is reduced by about 4 hertz. And we can show, using this modeling, that there's only one parameter in the model that changes during that drug administration, and that's the synaptic time constant on the inhibitory interneurons. And so it gets longer, the inhibitory interneuron time constant. And that fits really nicely with the literature from in vitro rat hippocampal slice cultures. So you put tiagabine on the slice cultures and you see a lengthening of the synaptic time constant. Okay, so I've gone way over time now, so I'm going to finish there. So what do I want to say in terms of summing up? So I think the first thing to say is that with MEG and EEG, it's possible with very simple and ultimately very boring experiments for your participants, you can measure very robust and repeatable markers of low-level oscillatory dynamics, which are tied, we think, very tightly to the underlying synaptic physiology in terms of this inhibitory interneuron pyramidal cell microcircuit. And that by the use of if it's physiologically informed modeling of that data, you can attempt to tap into specific, layer-specific and connection-specific modulations with drug and with diseases such as schizophrenia. So I'm going to stop there. Thank you, and apologies for running over. Yes. So Keith identified the fact that that in our model we identified only really one strong modulation, which was on the synaptic currents in the inhibitory interneuron, the time constant of that. Whereas it's known that tiagabine has a big effect on extrasynaptic, which are in the model. And this is obviously a key flaw in any model which doesn't include things that we know to be true physiologically. So the obvious thing to do there is to build a better model. So I'm not standing up here saying that this model is fantastic and everybody should use it and it's the end point. I think the way to think about the DCM in all its flavors, dynamic causal modeling, is as a framework that allows people like yourself to correct people like me and build in, and build in things so that the next time we analyze the data. So it's funny because naively when we started doing pharmaco-MEG work, I assumed that this moving away from a purely correlative approach was going to be fantastic because we could really find out how these gamma oscillations were. But of course, when you have something like tiagabine or any of the other drugs, they have a very non-specific effect on all the subpopulations of cells and synapses in the cortex, in visual cortex. So ultimately, it's quite difficult to figure out what's going on, even with these very pure drugs. But yes, good point. So the question was, with regard to the twin studies, could there be some shared environmental factors in terms that would move gamma?
It's so similar when they've done different things, right?
Yeah, I think the obvious inference there, although it is a big step, is that the very low-level synaptic physiology that we're talking about in V1 must be relatively preserved in terms of. I mean, we know it's plastic, and you can train it, and you can. There's sleep consolidation effects in primary visual cortex, for example. But if it's so heritable, most of the variants must be due to fixed effects, I guess.
Does it flip around with estrous cycle, for example?
Oh, now So we're asking about what comes and goes during the estrous cycle, sorry?
Synaptic, spine numbers go up and down.
Yeah, well, there's a few studies with GABA spectroscopy showing that GABA levels vary during the menstrual cycle, for example. And one of my interests is to try and tie measures of GABA measured with spectroscopy to oscillatory dynamics, and that with some success, but also some problems. So yes, I think the repeatability study that we did, people were tested every week for 5 weeks, and the measures were relatively stable in both men and women, and we didn't control for those cycles.